Selection of honeybees, honey yield and behaviour.
A young bee emerges from its cell. It will be one among 10 to 40,000 others which together form a colony of honeybees. Its survival depends on membership in such a community. Each colony has only one queen. She alone is capable of laying eggs and thus is the mother of all bees in a hive. Her attendants, the nurse bees, lavish intensive care on her, and this allows her to lay up to 2,000 eggs per day. The nurse bees also feed a glandular secretion to the larvae which hatch from the eggs, and in this way they exert an influence over the quality and quantity of the brood reared. Similarly, the expression of most of the characteristics of a colony is determined by the interaction between the queen and her workers. For its daily needs and winter stores, a colony collects annually about 25 kilograms of pollen and 200 kilograms of nectar. That quantity of nectar may be concentrated to about 70 kilograms of honey. Bees communicate with each other by means of a dance language. This tail-waggling forager is informing other workers about the direction and distance of a newly discovered source of food. Social food exchanges between them provide more information about the quality of the nectar. The construction of honeycomb is another example of the close cooperation among the workers of a colony. In order to produce wax, bees form dense chains during comb construction. In the form of tiny scales, the wax is secreted from specialized glands on the underside of their abdomen. The scales are removed with the mandibles and are worked to form an amazingly precise and strong cellular structure. Their brood is reared and honey may be stored. The overall productivity of a colony is therefore the sum of the manifold contributions made by queen, workers, as well as drones. In order to make optimum use of the varied hereditary characters in a breeding program, a study of the genetics and the mating process of honeybee queens is unavoidable. A virgin queen mates in free flight with as many as 20 drones. This is shown here in an experimental setup. The drone's semen is stored within the queen's reproductive organs, shown here in graphic simulation. At the tip of the abdomen is the sting chamber. It is separated from the median oviduct by the vaginal valve fold. Attached to the oviduct by a narrow channel is the spermatheca. The median oviduct forks into a right and left lateral oviduct. These lead to the two ovaries, which each consist of 120 to 160 ovarioles. The first drone mating with the queen forces his semen into the median oviduct. The mass of sperm of subsequent matings, only 3 more are shown here, force the semen into the lateral oviducts, dilating them considerably. After the mating, the queen compresses the semen back towards the vaginal valve fold. This makes 5 to 10% of the semen, up to 7 million sperms, enter the spermatheca. Thoroughly mixed, the semen is used to fertilize eggs over the next 2 to 4 years. Most of the semen is expelled as dry pellets. Just before an egg is laid, it receives a tiny droplet of the mixed semen. As many as 10 sperms may penetrate, but only one of them can fuse with the egg nucleus. Eggs laid in drone cells are an exception. They remain unfertilized. Multiple matings thus cause a brood of several groups of half-sisters, which all have the same mother, but are the daughters of several fathers. When drones with pronounced body markings had mated with a queen, their genetic differences express themselves in their daughters. Here, the genetic transmission of dark and of leather-colored abdominal shadings, as well as yellow rings, is evident. Furthermore, some workers have light, unpigmented eyes, while others have shortened wings. In spite of these complicated family differences, it is possible to breed honeybees. Of course, the composition and the relationships of the breeder colonies must be known, and their characteristics must be combined skillfully. All efforts of selective breeding must be directed towards producing a gentle, productive as well as disease-resistant bee.
Honig. Willst du mal lecken? Mhm. Dann mach mal mit dem Finger. Mach mal rein. So, guck mal. Okay, genug.
Apiaries in a variegated landscape with an adequate supply of food make ideal performance test bee yards. About 20 colonies should find sufficient forage all the year round. In order to emphasize the genetic differences between colonies, the management of all stocks must follow a uniform plan. Making hives face in different directions reduces drifting. Shook swarms or package bees of equal weight are highly suitable for setting up new testing colonies. Sister queens which had mated in the same mating apiary are introduced to all colonies to be tested within the same group. The queen cage is closed with a plug of candy and suspended between frames of foundation. After opening the swarm box, the brood chamber is placed on top. The bees will cluster around the caged queen within the next few hours. The lower hive body and the swarm box are removed later. Finally, the bees are given a supply of sugar syrup. All colonies in the apiary receive the same treatment. As an alternative, the young test queens can be introduced to established colonies. These should be healthy and of comparable strength. One method is the direct introduction by substitution, but this is only likely to be successful when the young queen produces eggs. Pheromonically, she is then of equal value to the bees and is usually well accepted. The old queen must be found first. She should have been free to move about in the hive until then. Had she been caged, the bees could have felt queenless and could have started to raise queen cells. That would endanger the acceptance of the new queen. The old queen is picked off the comb and is temporarily caged. Just before her introduction, the young queen is removed from her mating nucleus. Her number and markings are registered into the breeder's record book. For the time being, the old queen may be introduced to that mating nucleus until, after full evaluation of her data, it is possible to decide about her future use as a breeder queen. Over the next few hours, the bees will release the new queen after eating away the plug of candy. For a fair performance test, the introductions should be done before the end of July, so that a queen's inherent qualities, such as wintering and spring development, can be assessed in a colony consisting of her own progeny. All details of the queen's identity are entered on a record card, which will remain with her throughout the test. A first check is made after 5 to 7 days. The presence of eggs and very young larvae is evidence of a successful introduction. Colony strength is assessed in all stocks about 4 weeks after the introduction of new queens. Unequal bee populations could have been caused by differences in the past or maybe due to variable qualities of the bees in the shook swarms. Therefore, it is necessary to estimate colony strength as well as the areas of brood. Obvious differences are levelled out at once by the judicious removal or addition of brood. One frame of brood roughly corresponds to the number of bees which densely cover 2 frames. After this last operation, all colonies in the apiary should show comparable strength. Finally, all stocks are provided with sufficient stores for overwintering. Here it is done by adding the food chamber containing frames of honey. Any surplus spring honey is suitable for feeding. Shortages are then made up by feeding sugar syrup. All hives should show similar weights. The concentrated sugar syrup will be taken down within a few days and is stored in honeycombs. At the beginning of October, all stocks should have at least 20 kilograms of winter reserves. The first cursory spring examination is made when willow catkins are in flower. Food reserves, the presence of brood, and the health of bees are checked. As soon as colony strength begins to recover, the full evaluations are done at 3-week intervals. As each frame is removed from the hive, those covered by bees are counted. This is a very simple way of estimating colony strength. Any differences in development could be due to genetic factors, and this character could be important for the exploitation of certain crops. A thorough inspection of the brood nest is now made frame by frame. Every colony should have a frame for the construction of drone comb. In this case, the frame is subdivided so that some of the drone brood may be removed and distributed for breeding purposes. But no drone brood may be removed when the development of varroa populations is being investigated. When the queen is found, her identity is checked and she is examined for injuries. All breeder queens should therefore be marked for life with coloured and numbered discs. In order to assess the amount of brood, the combs with brood are counted, and additionally, the brood area of the central frames is measured. In this hive, the brood extends nearly into the corners of the frame, and the colony is awarded maximum points 4. For a fair evaluation, it is also necessary to look for brood gaps which may be due to inbreeding. This is made easier with a card with an opening which reveals precisely 100 cells of worker brood. The average of 3 to 5 counts made in early May represents the inbreeding factor. Another most important task is the regular evaluation of the colony's behavior. This requires extensive experience. This stock is very gentle and can be worked without smoke. The bees do not attack and do not sting. This is awarded top marks. After the inspection, all frames are returned in the original order and the hive is closed. All points awarded and other comments are entered on the record card. Some colonies can only be handled with full protection and with lots of smoke. Such defensive behavior is penalized in half-point steps, and this colony receives the lowest mark, 1. Another aspect of bee behavior is calmness on comb after its removal from the hive. When this character is pronounced, the bees form a dense covering on the comb. When the opposite character of running dominates, bees leave the exposed comb in a short time. Calm behavior is also evaluated with the 4-point scale. This patch of sealed brood shows many gaps. Some could have been caused when scattered pollen stores blocked egg-laying temporarily, but gaps to this extent are genetically linked and must be explained by the mechanism which determines sex in honeybees. About 11 different sex alleles are involved. Some of them are represented here as coloured symbols. Each fertilized egg has 2 such alleles. When they are unequal, it develops into a female, a worker bee, or a queen. Eggs which normally produce drones are, as a rule, unfertilized. They have only 1 sex allele. When the 2 factors coincide, a diploid drone larva, a freak male, hatches from the egg. It is removed and a gap occurs in the patch of brood. Within a few generations, inbreeding can lead to a brood nest with many gaps. Normally, the spermatheca of a queen contains many different types of alleles. Fertilized eggs have sex factors in unmatched pairs. Female larvae will develop. Now a virgin queen mates with the drones of her mother's colony. 75% of all eggs have unequal alleles, while 25% have identical sex factors. So 25% brood gaps occur. When the granddaughter mates with drones from the grandmother's colony, half of all eggs laid possess alleles of unmatched types, while the other half has identical alleles. A brood loss of 50% results. During the swarming period, it is necessary to make weekly examinations of the colonies, apart from the regular visits for full evaluations. Frames are scrutinized for queen cells. When any are found, they are opened. In this case, they contain neither eggs nor larvae, and the colony is not in a swarming mood. If the first honey super is partially filled, the stock is given another one. This super contains frames with foundation and drawn comb. It is weighed and its empty weight is entered in the record book. This colony has swarm queen cells already containing larvae. A large number of swarm cells with well-provisioned older larvae are a sure sign that the colony is determined to swarm. All cells are destroyed. Whenever the swarming fever is that far advanced, it is not possible to suppress it by adding supers or by the destruction of queen cells. Swarming can only be controlled by making a temporary parking nucleus with the old queen. After lifting off the upper brood chamber, all frames are carefully examined and all queen cells are removed. The marginal areas of drone combs should not be neglected either. In preparation for the next step, one frame of open brood is selected and put into a spare hive body in which the other frames contain foundation and drawn comb. Meanwhile, the other brood chamber has been examined for queen cells. The hive is now reassembled in the following way. The new brood chamber with the single frame of brood is placed on the original floorboard and the honey super on top. This is the queenless part of the colony, and all flying bees will soon return to it. A crown board separates both parts of the colony. The queen right brood chambers are put on top. All flying bees will return to the familiar original hive entrance over the next few hours. This weakens the queen-right nucleus to such an extent that its swarming fever is extinguished. After 7 to 9 days, the emergency queen cells, which have been raised on the single frame of open brood in the lower hive body, are destroyed. Another frame of open brood is introduced in its place. This keeps bees content and avoids losses through drifting. The two halves of the colony are reunited after a further 7 to 9 days. The swarming tendency is extinguished and the queen shows full laying activity. The hive is reassembled in the original way. Once again, any emergency cells found on the single comb of brood must be destroyed beforehand. This method of swarm control permits the continued evaluation of the queen and of her colony. Naturally, the control of honey production is the paramount consideration. It can be ascertained by frequent weighing of colonies during specific nectar flows. Any changes in the colony must of course be taken into consideration, such as additional supers. The use of clearer boards and bee escapes simplifies the removal of the honey crop. One day before supers are collected, the clearer boards are put in position. Bee escapes allow trapped workers to pass down into the brood chamber but prevent their return to the supers. Supers can be collected the next day. The honey yield is established by weighing the supers. The origin of the queen and the purity of her matings is checked by testing her progeny morphometrically. In order to make quite certain that only the queen's own descendants are sampled, only freshly emerged bees and drones are taken off the frames. About 50 bees and 50 drones are required for a full assessment. Because the chitin of the young bees is still too soft, they are held in cages for a few more days before the samples are preserved for a later examination. Another aspect of performance testing is the selection of vital and disease-resistant stocks. Only they can produce optimum results. All data comprising honey production, gentleness, calm behavior and swarming propensity are assessed at the end of the season. Those stocks which had been in the same testing station can at once be compared, provided the original queens were present at the end of the test period. Under the conditions of fair performance testing, the colonies show their differing qualities. Most characters are distributed normally. This means that very few individuals score very high or very low marks, while most fall into the category of average. Selection means choosing the best individuals for further propagation. The selection difference, the superiority of a character over and above the average, depends on its scatter within the population, as well as on the intensity of selection. Heredity determines to what extent the selection difference is passed on to the next generation as an improvement. With continued selection, the gene pool of a population grows more and more uniform. The possibility of further improvement diminishes with subsequent generations. Yet, the level of performance of a thoroughly selected population establishes itself at a high plane, as long as inbreeding causes no damage. The continuous and collective work of selective breeding has improved the performance and character of our honeybee considerably. The task of selecting for varroa-tolerant bees confronted breeders with a new challenge. The successes achieved in selective breeding, especially for pronounced gentleness, allowing colonies to be kept in densely populated areas, can only be maintained through ongoing selection.